Pholin! Welcome to the Poblacion's Candy Factory. Do you want to know how our candies are produced? Well, you just met the right person for the task. Come, I'll show you around. So, here's the boss giving the recipes to the messenger. Then, the messenger leaves the office through the door and goes to the factory floor and workstations. The workers pick up the ingredients. Here, you can see that the workers assemble the ingredients at the workstation. Ingredients are combined according to the recipe. The candy assembly is finished. Workers eat some, rest is wrapped from shipment. Easy as that. Well, before you leave, I heard that you are having a hard time learning about the protein synthesis. I can help you with that. By way of analogy, our candies are being built in many simple steps, like what I exactly showed you earlier. Protein production or protein synthesis is similar to candy production. How though? Well, let me discuss the steps again one by one. The boss gives the recipe to the messenger. In this scenario, it represents how DNA gives instructions on how protein is assembled through the messenger, which represents mRNA. What is messenger RNA? Messenger RNA or mRNA brings information from the DNA in the nucleus to the protein manufacturing area. To sum up this process, DNA serves as the template in making a messenger RNA strand. But how exactly is mRNA made and how does it move to the manufacturing area? I'm about to explain to you the first process in protein synthesis, the transcription. Ribonucleic acid polymerase enzyme binds and opens the DNA molecule that will be transcribed. As the DNA molecule opens, the RNA polymerase slides along the DNA strand and links free RNA nucleotides that pair with the nitrogenous bases of the complementary DNA strand. Now, take note of this. These are important. Nitrogenous bases have two types. Purine bases, which are adenine and guanine. Then we have the pyrimidine bases, which are the cytosine and thymine on DNA. And we have cytosine and uracil on RNA. Right here is the complementary base pairs of DNA strand to another DNA strand. And right here is the complementary base pairs of a DNA strand to an RNA strand. Now, let's go back. If the sequence of bases on the DNA strand were CCG, TTA, CAT, the sequence of bases on the RNA strand would be GGC, AAU, GUA. As you can hear, I am emphasizing the presence of U or the uracil because as you can see here, we are transcribing the adenine to uracil instead of thymine. Why? Because RNAs do not have thymine. Now, the next step. The messenger leaves the office through the door and goes to the factory floor and workstations. Just like the mRNA, when the process of base pairing is completed, the mRNA breaks away as the DNA strands rejoin. The mRNA leaves the nucleus through a nuclear pore and goes to the cytoplasm. So, here, the door is the nuclear pore and the workstation is the cytoplasm. And this is how the transcription process ends. But the protein synthesis doesn't end there because we're still halfway in making our candy or the protein. Now that we have the mRNA, its order of bases determines the protein synthesis. This is where the process of translation happens. In translation, each set of 3 nucleotides in an mRNA molecule codes for 1 amino acid in a protein. This explains why each set of 3 nucleotides in the mRNA is called a codon. Each codon specifies a particular amino acid. For example, the first codon, which is cytosine, guanine, and uracil, instructs the ribosome to put the amino acid arginine in the protein. The sequence of codons in the mRNA determines the sequence of amino acids in the protein. But how are the right amino acids added in the right sequence to match the sequence of codons in the mRNA? As translation begins, mRNA binds to a ribosome. Going back to the factory, the workers pick up the ingredients like how transfer RNA binds to amino acids. So, the tRNA molecules, each carrying a specific amino acid, approach the ribosome. Like how the workers assemble the ingredients at the workstation, the tRNA anticodon pairs with the first mRNA, codon, arginine, uracil, and guanine to form the initiation complex. The two molecules temporarily join together. Usually, the first codon on mRNA is AUG, which codes for the amino acid methionine. For you to remember AUG easily, you can just think of August. AUG signals the start of protein synthesis. Then, the ribosomes slides along the mRNA to the next codon. When the first and second amino acids are in place, an enzyme joins them by forming a peptide bond between them. As the process continues, a chain of amino acids is formed until the ribosome reaches a stop codon. So down here are examples of stop codon on the mRNA strand. This is similar to how ingredients of the candy are combined according to the recipe, like a polypeptide chain grows in response to mRNA codons. And there we go! Candy assembly is finished. Workers eat some of it and the rest is wrapped for shipment. Just like how the polypeptide chain is released and formed into a protein. The protein is either used by the cell or is packaged and exported outside the cell. And this is when the protein synthesis is complete. I hope you learned a lot. But if you want to have a further understanding about the protein synthesis, stay tuned as I will explain to you a more detailed process starting from the nucleotide structures and components. For now, let's test your understanding about our topic by answering these following questions. You can stop the video. Now don't forget to comment your answer, and thank you for watching and see you next week. Bye!